So today we're going to talk about protein. Protein play a very important role in all life process. So protein account for about 50% of the organic material in the typical animal's body. So this table, I'll list a few selection of a protein which are very common in nature. So we're going to categorize it by its function. Like, for example, the first category, which is very important, it's proteins involved in gene expression and regulation. What's its function? It can make mRNA from a DNA template, synthesize a polypeptide from mRNA, can regulate the genes. So one of the example of this type of protein is RNA polymerase, which can catalyze the synthesis of RNA, use DNA as a template. The second one is motor protein, so it can initiate the movement. Christine, stop me anytime if you have any question, okay? Otherwise, I'll keep going. Okay, one of the example is myosin, provide the contractor for force of muscle. And then the third one is defense proteins. It's protect the organism against disease. A good example is antibodies help destroy bacteria and virus. And the fourth one is metabolic enzymes. It's increased rates of chemical reactions. One of the example is hexokinase is an enzyme involved in sugar metabolism. The fifth one is cell signaling protein. It enables cells to communicate with each other. And one of the example is taste receptor in the tongue, allow animals to taste the molecule in food. And then another one is called structural protein. It support and strength structures. Like for example, actin provide shape to the cytoplasm of the plant and the animal cells. And the last one is called transporter protein. It's promote the movement of solids across membranes. Example is glucose transporter, move glucose from outside cell into the inside of cell. Then we're gonna talk about the building blocks of amino acid. So, amino acid is building block of a protein. So, protein are composed of carbon, hydrogen, oxygen, and nitrogen, and maybe a small amount of like sulfur. The monomer of protein is amino acid, like component with a structure in which a carbon atom called alpha carbon is linked to amino group. Like this is a good example about the structure of amino acid. So it's carbon in the middle, and then there's 4 bonds around it. So one of it, like there are like 21 most common amino acid in the nature, and then all of them like can simplified by this structure. So this amine group, carboxyl group here and the proton here, the only difference between these 21 amino acid is this R group. Okay, so and you can tell this carbon center is a stereo center. So this compound will have stereoisomers. So the most times the amino acid in the nature is alpha carbon. So which means this stereo center on the carbon, it's already identified. It's only in one type, not the beta, only alpha. So like for example, most of the times this amine group in the water would be protonated, which means this amine group can accept the hydrogen ion, and then this will be partially charged. And this carbolic acid group, it's easy to lose 1 proton from this OH and become negatively charged. So all amino acids except the glycine exist in more than 1 isomeric form. What's that mean? Like for example, glycine is the only one this R is equal to hydrogen. If this is like a hydrogen, that would be like 2 hydrogen here. So this carbon is not the stereo center. And the rest of the amino acid, this is R is not H. So which means they are bonded by the 4 different functional group. So this carbon is stereo center. That's why this structure has isomers, stereoisomers called D and L forms. And these D and L forms, they are enantiomers. Only L amino acids are found in the protein. D amino acids are not found in most cells. That's an exception, like for certain bacteria cell wall, where D amino acid will play a protective role against the molecules secreted by the host organism in which the bacteria live. So remember, in the nature, most of it is L amino acid. So this chart lists the most common 20 amino acids. You can tell the difference between these 20 amino acids is about their side chain, which is our group here. So the only exception is glycine. Hold on, where is glycine? Oh, see, this is glycine. So this is the R is H. So let's look at it one by one. This is called aniline. So R is methanol group. This is valine. So R is the isopropyl group. And then leucine, this is like a 4 carbon chain. And the isoleucine, there's a 4 too. But this is methyl group. It's on the first carbon. It's not on the second carbon. And then proline, this is like cyclic. This is a ring formed. And then phenylalanine, phenyl, which means one of the phenyl group is here. And then tryptophan, okay? This is some kind of double ring replace the phenyl. And then there's cysteine. So this is a sulfur here. And then this one is methionine. So this is like 2 carbon, 1 sulfur, and then the methyl attached to sulfur. Serine. So instead of sulfur compared to cysteine, this is OH. Sorry. Threonine. This is like OH is here. Asparagine. So you can see this is what's this group called? This is called amide, which is COO and NH. It's called amide. The glutamine. This is very similar to asparagine, but 1 more carbon compared to the asparagine in our group. This is the tyrosine. So this is like phenol. It's called the phenol, the OH phenol group here. And then this is aspartic acid. So this is like carbonic acid attached to the CH2. Glutamic acid. This is another carbonic acid here. And then histidine. This is some kind of fiber membrane, but in this fiber membrane, there are 2 nitrogen here. And then lysine, that would be like long chain with amine at the end. Arginine, okay, is at the end. It's such kind of a group. So you should read about it very often. Try to remember as much as possible. So basically, this 20 amino acid can be divided into 3 groups. This is nonpolar. What I mean by nonpolar, because of the side chain group are like CH bond, those kind of things, or SH bond. Okay, it is nonpolar. Why is like a polar but uncharged? Why is polar? Because they always have like hydroxyl group on these kind of carbonyl groups, or this is like a hydroxyl group. Like this type of bond are polar, and this is polar charge. What I'm saying is charged because it's either carbonic acid attached to it, or like amine attached to it. Okay, this is amine. This is a carbonic acid. So the arrangement and chemical features of the side chain cause protein to fold and adopt their 3-dimensional shapes. So basically, So like for example, protein is it's a three-dimensional structure. Like they have a different structure of the protein. Sometimes it's called alpha helix, beta sheet, those kind of thing. But why causing different protein have a different structure? Because of this kind of composition of individual amino acid. Because of the this amino acid, when they combine into a protein, they will have some kind of interaction between each other. And then this kind of interaction between each other causes a different shape of the protein. Like I said, so certain amino acid side chain may be critical in protein function. For example, amino acid side chain found within the active site of enzyme are important in catalyzing a chemical reaction. So this amino acid, this 20 amino acid, it's a basic unit of a protein. It is because of the composition of the amino acid in each protein. It's decide protein structure. It decide of the proteins function. So when you want to oversee the big protein's function and structure, always go back to small scale, which is amino acid. So polypeptide are linear sequence of amino acid. So how this protein, like for example, then we have to think about how this kind of amino acid form the protein. That's like protein is like longer chain of amino acid. Like polypeptide is the shorter chain of the amino acid. So polypeptide and the protein are all polymer of amino acid. The difference between polypeptide and the protein is the length. It's the number of amino acid with inside. So that's what I'm saying. Amino acids are joined together by a dehydration reaction that links the carbonyl group of one amino acid to the amine group of another one. So what that mean is, like for example, this is one amino acid, glycine, and then this is another amino acid called alanine. So like for example, carboxyl group of the glycine will react with the amine group on the alanine, and then it will form those kind of CH bond. So basically, how it formed? It will form like lose 2 hydrogen here and 1 oxygen here, and break these 3 bonds. So basically, this H2, 2H, and the oxygen will be losing the water. That's why it is called a dehydration reaction, because it is lose 1 molecule of water. So that's form this kind of CN bond. And then there's like CO here, and then N here. This is called amide functional group. So we can call this kind of bond called a peptide bond, right? This covalent bond. So this is one of the example. Like for example, so this these two amino acids linkage can extend either from this left side or from right side. For example, amine in here will connect with another like carboxyl group of another amino acid. It extends, and then carboxyl group on this end will connect to another amine group of another amino acid. So on and on, and then this large chain will formed, and then this is called a polypeptide because this is one peptide formed by two condensation of the 2 amino acid. Then there's like multiple polypeptide like this. It's called a polypeptide. Okay, so the backbone is like this, always CN, right? Multiple peptide bond. And then at the end, there always be like 2 ends left. Like for example, this end would have like NH group from from this amino acid, and on the other end we have a free carboxyl group. So we can say this is N-terminal, because why it's called N-terminal, which because it has a mean functional group here. Why it is called a carboxyl group? Why it is called C-terminal? Because it has a free carboxyl group here. Okay. So polypeptide refers to a structural unit composed of a linear sequence of amino acids. A protein is a functional unit composed of one or more polypeptides that have folded and twisted into a precise three-dimensional shape. So which means polypeptide is short, and then protein is like multiple such type of polypeptide formed together, like into a large structure, and then it can fold, can twist it, and then become like a three-dimensional shape. So one thing I should mention about some, like most of the times protein is not only like a polypeptide composition. It can be like only it's like polypeptide chain. There are always some kind of modification. Sometimes it can modified by carbohydrate. That's called a glycoprotein. Why it's called a glycoprotein? Because glyco represented glycan. Glycan is the same thing as carbohydrate. Another one is like this kind of polypeptide chain. Polypeptide chain. It have a lipid like modified somewhere. It's called a lipoprotein, because attached to a certain amino acid, like for example, maybe some kind of a carbohydrate can link to here somewhere. It's just a link to the side chain, and the same thing like lipid can link to the side chain too. So this type of modification impact the unique function to such proteins. So protein have a hierarchy of structure. And what's that mean? So like, for example, protein have a four progressive level: primary structure, secondary structure, tertiary structure, and quaternary structure. Let's see each type of a structure one by one. What is primary structure? Primary structure just a linear sequence of amino acid. What that mean? When you have a protein, you just check the composition of this protein by its individual amino acid. So like, for example, have MPTLH, something like that. This is primary structure. What's called secondary structure? Certain sequence of amino acid form the hydrogen bond that cause the region to fold into a spiral or sheet. What's that mean? So like, for example, this is like based on the primary structure. This is like linear linear amino acid, but then it's not like a thread. It's not like this thread, because there's some kind of interaction here. Like, for example, this carbonyl will formed the hydrogen bond with like another, maybe let's say this is like amino acid 1, this is 2, this is 3, this is 4. No, so the oxygen from the first amino acid will form the hydrogen bond with the hydrogen on the 5th amino acid, right? Now, so this kind of bonding, there's a more. Like, for example, the second amino acid oxygen will form the hydrogen bond with, let's say, this 5, this 6 like the hydrogen from the 6th amino acid. It's like this, so on and so on. And then instead of a linear one dimensional, will have like this kind of, it's called alpha helix. It's become like a 3-dimensional alpha helix. The same thing would be here. Like, for example, beta sheet. Why I'm saying it's beta sheet is because, like, for example, this is kind of linear, okay? It's like turn around, like this this sheet is like a piece of paper. This piece of paper are composed of these several amino acids, and then this sheet are composed of this type of amino acid, and then in these two sheets, they have some kind of hydrogen bonding between each other. That's called a beta sheet. Okay, it's all because of hydrogen bonding. So tertiary structure, then the further on, like higher level, it's called a tertiary structure. So which means secondary structure and the random coil region fold in the three-dimensional shape. What's that mean? It's like, for example, this is the alpha helix, and then this is the this is like a random coil. Random coil. It's neither a alpha helix or beta sheet, but it's because of this kind of random coil region, random coil things. It's connect with this part of alpha helix to the beta sheet here. Okay, and then another random coil connect this beta sheet with this alpha helix. Keep going. Okay, this another one, random coils. That's a tons of a random coil here. Like for example, this random coil connect this beta sheet and with this beta sheet. So this become like a three-dimensional tertiary structure. Okay, and then there's one more level that called a quaternary structure. Two or more polypeptides may bind to each other to form a functional protein. Like for example, so this blue one, it's a long chain, polytrans. It's an individual one. And then this one is another individual. But this blue one and this with this green one, they are not connected together. They just like they're individual, but like just like two things, they are separate things. They're not connected together. But because of some kind of like side chain hydrogen bonding or some other interaction, most times the hydrogen bonding, they just bond together like this. Two things just bind together. That's called quaternary structure. So this is very important to remember. So like, so let's talk about the each level of structure in detail. First of all, let's talk about like a primary structure. It's a protein, is an amino acid sequence of its polypeptide. So the primary structure of a protein are decided by the genes, by the DNA. So we will explore that information in the chapter 2. So gene carries all the information for the production of a polypeptide with specific amino acid sequence. So basically, like for example, this is a large proteins. In large protein, they have like 122 amino acid. Throughout from amino acid 1 to 124, what it is, which amino acid it's from this 20 amino acid, it's all decided by the gene. Okay. So like for example, this is protein called the ribonuclease structure. That's a primary structure. It's give you the detailed linearity amino acid. What's the function of a ribonuclease? It can act as an enzyme to degrade the RNA molecule after they no longer used by our cells. So you can tell this ribonuclease, it's composed of the very short polypeptide. It's only have 122 amino acid, like 122 amino acid. Let's say like very short amino acid. So average protein has about 300 to 500 amino acid. Some protein have like a few thousand of amino acid long. And then let's talk about the secondary structure. So the so like I said, the amino acid sequence of polypeptide cause polypeptide fold into a more compact structure. So like amino acid can rotate around the bond within the polypeptide. So which means like, for example, between the C and H, this bond is single bond. It can rotate. Like for example, if you rotate 100 degree back, then this O will be up. So this kind of composition will not form the hydrogen bond with the NH here. But everything it's favored energy like lower energy configuration, because on the like when you rotate it 100-degree back, this oxygen will be downward. It will form the hydrogen bond with the NH here. So because this kind of alpha helix, it's from the energy perspective, it's lower. So by forming this kind of hydrogen bonding, so I'm saying the triangle are very flexible, can fold into the different shape. So folding can be irregular, or certain region can have a repeating folding pattern called secondary structure. So the two basic type of secondary structure are alpha helix and the beta sheet. So I already explained alpha and beta. You can read it by yourself. Tertiary structure. So I already talk about her. Okay. So let's talk about the protein structure. It's determined by several factors. Like for example, you have like gene decided the primary structure, but secondary, tertiary, and quaternary structure are decided by basically it's by structure, but determined by several factors. It's like hydrogen bonding. It's the very common ones we see, right? And then another one is ionic bond and other polar interaction. So like for example, within this secondary, tertiary, and quaternary structures, beside hydrogen bond, I just show you there's a kind of other interaction between the amino acid. There are some ionic bond and the polar interaction, because that amino acid chain has positive and negative charge. Like for example, I mentioned before in this big chart, there's some third type of polar amino acid, right? All these carbonic acid amine, they're all either positive charge or negative charge. So this kind of positive and negative charged group, they can interact with each other, right? Forming some kind of ionic bond. And then uncharged polar side chains in a protein may bind to ionic amino acid. This is by polar interaction, like polar side chain, but they are charged, can also form the polar interaction with ionic bond. So this type of ionic bond and polar interaction, it's all like interaction between a polar functional group. Another thing is called hydrophobic in fact. What is called hydrophobic interact? It's because of like some amino acid side chain, they are non-polar. So let's first introduce the concept of hydrophobic. What is hydrophobic? Hydrophobic, which means this functional group is non-polar. So the non-polar, there are some kind of interaction between the non-polar functional group. Even so, those kind of interaction are much weaker than the ionic interaction and the polar interaction, but they do exist. Like for example, as a protein folds, the non-polar amino acid are likely to be found in the center of the protein, minimizing their contact with water. Like for example, because the protein most times it dissolves in the water, but then it has a structure in it. Outside is the water, so water likes polar functional group. So polar amino acid will be outside exposed to the water environment, and the only those kind of non-polar amino acid, they will aggregate. They will aggregate in the middle, right? This is how the protein structure. Like outside is hydrophilic, it likes water, so it's outside. Hydrophobic, it's nonpolar. It's hate water, so it's segregated in the middle, which can help them to get away from the water. Some protein has stretches of nonpolar amino acid that anchor the protein in the hydrophobic portion of the membranes. Okay, what's this mean? Means like because membrane, when I'm talking about the lipid before, you know, like lipid have two layers, and then on the top layer and the bottom layer, it's a phosphate group, which is polar, and then in the middle there would be like a lipid, like a tail, aggregated in the middle. So in the middle part, that's the nonpolar. So what this means is like nonpolar amino acid will be merged into the inside, the middle part of the membrane, because in the middle, that part, that's also like hydrophobic. So hydrophobic nonpolar things, they like attracting each other by hydrophobic interaction. And then another interaction we should talk about is van der Waals dispersion force. What's the van der Waals dispersion force? Like atoms within the molecule have temporary weak attraction for each other if they are optimal distance apart. So basically, atoms within the molecule, they attract each other, they attract each other very weak, but even though this kind of interaction is weak, it do exist. So this type of like weak attraction, it's called van der Waals dispersion force. So it has to be in optimal distance apart. So like, for example, what that means is like, if these two columns are too close, their electron cloud will be repel each other. Then they don't have those kind of a van der Waals dispersion force. When they are apart, they are far apart, they have to be in like a certain range. It cannot be too close, they repel each other. It cannot be too far away. Those kind of van der Waals dispersion force will disappear only in some certain distance. So this type of van der Waals dispersion force contribute to the tertiary structure and the quaternary structure. And another type of very important interaction between within a structure is called disulfide bridge. What's that mean? Like, for example, this is a long polypeptide chain, right? And then here it's a cysteine, because cysteine in each side chain, it have a sulfur SH functional group. And then there's another cysteine here, has another SH. When this two SH functional group, when they meet each other, there was some kind of reaction going on. So these two sulfur will form the bond and lose these two hydrogen. This is called disulfide bridge. See, because of this kind of a bond formed, so these two part, they're very close with each other. So it's changing the configuration, the three-dimensional structure about the protein, right? This kind of sulfur disulfide bond also important in protein folding, because the covalent nature of the disulfide bond can help to stabilize the structure of the protein, right? If we don't have this kind of sulfur bond, and then this strand will be far away from this strand, or it will be very loose, right? And then when they bond together, the structure of protein, it's more rigid because there's another bridge here. So here we say this first, okay. The first four factors, like hydrogen bonding, van der Waals, ionic bonding, and hydrophobic. In fact, important in the ability of different proteins to interact with each other. Many cellular processes involve the steps in which two or more different proteins interact with each other. What that mean? So like two proteins, like protein have to interact with each other for a biological process, right? That's very, very important. It needs to have some kind of interaction going on for chemistry going on in the body. So how and why these two proteins can interact, because there's like either hydrogen bond, hydrophobic effect, or van der Waals effect, or ionic bond, or poly bond. Those kind of effect interaction bring these two proteins together, and then something can happen. So when such kind of interaction happened, one protein must recognize and bind to the surface of the other. Such binding is very specific. So the surface of one protein precisely fit into the surface of another. Like for example, what does that mean? It's fitting. Like for example, it's like a puzzle. This part have to fit in here, right? This part can fit in here. It's like a puzzle. It's like a puzzle. When you play the puzzles, one piece have to fit with another piece. That's what it means. Such protein and protein interaction are very important in allowing cellular process to occur in a series of defined steps. And then these protein and protein interactions are very important in building complicated cellular structure, can provide shape and organization of the cells. So one thing you should take from here is this kind of interaction has to be specific. Like for example, it's like a puzzle. This protein has to be fitting with this one, right? Because it have to fit here, this one have to fit here. It's like puzzle, they have to fit together. Cause this kind of specific interaction. Like for example, if some other piece of a protein doesn't have this kind of shape, it won't fit in this glove. It's called a glove. And then interaction doesn't happen. Now chemistry won't happen between these proteins. Okay. You can read about this. Let's talk about the nucleic acid. Okay, nucleic acid are organic micromolecule that account for only about 200 weight of animal like humans. But even though it exists in very small amount, but it's extremely important because they are responsible for the storage, expression, and the transmission of genetic information. So the genetic information exists in the nucleic acid. In genetic information are all existing in the nucleic acid. That's why it's important. Why this gene is important? Because expression of genetic information in the form of specific protein decide which category the organism belong to. Like for example, like human being and the frog and the bacteria. Why we are human being and why this animal is a frog? Why this organism is a bacteria? That's because they have a different protein, but where's the protein coming from? It's from genetic information. So this kind of a nucleic acid decide which organism it is. So most commonly, nucleic acid have two categories. It's called DNA and RNA. Okay, let's first talk about the nucleic acid, DNA and RNA, its structure. So so both DNA and RNA, they are polymers. Okay, if they are polymers, there must be like a building blocks for both of them. So DNA is building block. Okay. First, let's talk about DNA. What's called the DNA? It's called the deoxyribonucleic nucleic acid. RNA, it's ribonucleic acid. Okay. So DNA has store genetic information. RNA will involved in decoding the information into instruction for linking a specific sequence of amino acid. So DNA, so when we talk about gene is DNA. What's RNA for? RNA help translate DNA into the protein. Okay, so the building block for both DNA and RNA called a nucleotide, right? What's nucleotide? This is a good example of a nucleotide. Okay, so nucleotide have two types. DNA, it's nucleotide. It's called deoxyribonucleotide. Okay, and then RNA, it's nucleotide monomer. It's building block called ribonucleotide. Let's talk about the more details about this. Like for example, DNA building block, deoxyribonucleotide. It have 5 membrane, have a base and a phosphate. Okay, and then the same thing for the ribonuclease. It has 5 membrane, it has a base, it has a phosphate. But pay attention, the difference between the ribonucleotide and the deoxynucleotide is this 5 membrane. Why? Because deoxynucleotide only have OH in this is carbon 1, 2, 3. Only have OH in the carbon 3. Okay, it doesn't have. But ribonucleotide, it have hydroxyl group in both C2 and C3. That's why we are saying deoxy. What do you mean deoxy? Get rid of 1 oxy, right? Compared to 2 OH here. It only have 1 OH here. That's called the deoxy because it's missing one of the oxy. And another thing is mentioned about, that's one of the difference between the deoxy and the ribonucleotide because of the number of OH group in the 5 membrane. Another thing I should mention about is the base. So in deoxyribonuclease, they only have A, T, C, G. These 4, but in the ribonuclease, they don't have T, they have U instead. Okay, let's be very careful. So okay, so first let's put it in simple ways. So the base here are 2 types. They like A and G called purine, which have a fused double ring. Okay, it has 2 rings together. So in this chart, this is called a pyramid. Why? Because it have only 1 ring. That's we have structure here. Okay, for example, this is called the purine A and G, right? They have 2 rings. So, but pay attention. What's the difference between A and G? See, this ring, this 5 membrane ring is the same. The difference is about this ring. This is NH, but this become CO, carboxyl group, right? And then this is NH, is H. So the difference between this functional group and this functional group. Okay, that's called this called purine because they have 2 rings together. And then this is called this C and T. This is called the pyramid. C and G called the pyramid. It's different from purine because it only have 1 ring, right? Instead of 2 ring here. And then the difference between the C and G is very similar. This have a amine, this have a CO, okay? This, okay. Another thing is this one have a methyl, this have a OH. Okay, so So this is for DNA. Remember, DNA have A, G, C, T, those two base. Okay, so when we're talking about DNA nucleotide, base only have 4 base. So which means DNA only have four types of building block instead of like amino acid. Amino acid have 20, and then but gene DNA only have 4 types. Sorry. So remember when you have to label it, this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Okay, and then DNA molecule consists of 2 strands of nucleotide coiled around each other to form a double helix. What that mean is this. So DNA have 2, like, for example, think about like amino acid. Amino acid is like one, if we make it straight, like big straight, one line, right? It's one line. But DNA have 2 lines, like 2 linear of nucleotide, right? Like they twist it together. How they can bond together? It's by like, for example, this is one strand, this is another strand, and then they twist together to form this kind of a alpha helix, double strand. That's called double strand by this kind of hydrogen bonding, right? The base from one strand will have hydrogen bonding with the base of another strand. Okay. As I'm saying, the 2 strands hold together by hydrogen bond between a purine base in one strand and a pyrimidine base. So what's that mean? So this is a pyramid because it's double one ring, and then this is a purine as a future double ring. So one thing you should remember, only certain base can pair with each other due to the locations of hydrogen bonding groups in the 4 base. In a DNA molecule, A on one strand is always paired with T on the opposite strand. So A only pair with T. A cannot pair with the G and the C, only with T. G is always paired with C. So keep that in your mind. A always pair with T. So which means what that means? Like for example, this is T, right? So in the another strand, it's when it's crossed with each other, that's always be A. It cannot be C, cannot be G. Okay, A and G they pair. And then if this is C in one strand, and then in other strand must be G, cannot be A and T. Okay, A and T, C and G. Remember that. Remember these pairing tools. So based on this kind of information, A and T and C and G, we can calculate the amount. Like for example, if a DNA molecule is composed of 20% of A, which means it also have 20% of T. If it have 60% of G, then C would also be 60%. Because C and G, they are pair, they should be equal. And that's how it. And RNA structure, let's talk about RNA. RNA structure only different in a few respect from the DNA structure. Like DNA, RNA consists of nucleotide covalently linked together. But RNA usually consists of a single strand of nucleotide. What's that mean? This is DNA, it's double strand like this, right? But instead, RNA it's just a single strand. Have one strand, doesn't have a double strand. So in RNA, sugar, like 5 membrane in the ribose, it's the ribose instead of deoxyribose. And then another thing I should mention about, in the RNA, we don't have a T. We replaced with U, right? Like we are saying A paired to T. There's no T existing in the RNA, only the uracil. That's so, but AGC would be the same, right? So in DNA, they have ATCG, but in RNA, only have AUCG. We finish this chapter. Let's go to the next one. Cell. So in this new unit, we're going to talk about the structure of synthesis of cell membrane and how the substance can be transport between the two phase, between the two phase of cell membranes. And then we're going to talk about metabolism, the chemical reaction in a cell and the organism. And then we're also going to talk about the enzymes, how enzyme can facilitate a chemical reaction. And then we're going to focus, then we're going to talk about how carbohydrate break down and generate ATP. And we're also talking about the photosynthesis. And then how the plants can get energy from the sunlight and convert this energy from sunlight into the carbohydrate. Then we're also going to talk about how cells interact with the environment and each other. And then we're going to talk about how cells respond to the signal. And last, we're going to talk about how cells interact with each other, produce a multicellular organism. And first, we're going to talk about the cell and its structure. Okay. Let's talk about arranging of the living cell on Earth. So living cells are very complicated connection of a molecule and micromolecule. So like, for example, it's a single cell. You think that's cell. A single cell is very small. You cannot see it from naked eyes. Most times you have to take advantage of the microscope can see the cells. You think that's small is simple, but it has a lot of a molecule, a small molecule and a micro molecule inside. It has RNA, DNA, protein, all kind of molecule and micromolecule inside. Like for example, a cell has to live. It needs DNA to store the genetic information, and then it needs RNA to help convert the genetic information into the protein, and RNA, beside the help to generate the protein, RNA also play other important roles. And it also has the protein, like this big molecule within the living cells. And then, and the proteins forms the foundation for structure and the activity of living cells. So living cells, it requires the interaction between the DNA, RNA, and the protein for its existence. So we're going to talk about how life gets started. So this one, it talk about the several stage for cell processing from the original cell. Stage 1 would be nucleotide and amino acid were produced prior to the existence of the cells. Like for example, before there's a cell existing in the earth, we have to connect those kind of basic foundation, basic molecule like DNA, RNA, and protein. So DNA, it's a nucleotide, right? Protein is amino acid. So this kind of building block, a monomer have to exist first. So that's why stage 1 is nucleotide and amino acid produced, right? Second stage, it's form the RNA and the DNA. So which means nucleotide will form the RNA and the DNA. Amino acid will become a protein, right? In the third stage. And then all these molecules, RNA and the DNA and the protein, have to encode in the cell membrane. And then on the fourth stage, those kind of polymers, which are enclosed in the membrane, acquire the property that's associated with living cells through all kind of chemical reaction. So, which means in the last stage, all this DNA and the RNA and the proteins, they interact with each other, which make a cell life. Then let's talk about the stage 1 by 1. Then let's talk about the stage 1. Organic molecule forms prior to the existence of the cells. So that's all like hypotheses here. Some like Russian biochemists, they propose that organic molecules such as nucleotide, amino acid arose spontaneously under the condition that occurred on the early Earth. According to their hypothesis, the spontaneous appearance of organic molecule produced what they called a primordial soul, right? So which eventually give rise to living cells. So there's a lot of hypotheses here. Some current hypotheses suggest that organic molecule like nucleotide and amino acid, they formed spontaneously. And those type of organic molecules slowly accumulate because there was little free oxygen gas. So they were not spontaneously oxidized. And there were as yet no living organism, so they were not metabolized. So what's that mean? Is this type of like nucleotide amino acid, they formed spontaneous because back then, like a long time ago, so there's no oxygen in the earth, so they cannot be oxidized. If they are oxidized, it's gonna be destroyed very easily, and there's no living organism back then, so they cannot be metabolized. So because they are very stable, they like slowly accumulate in the early ocean over a long period of time, called form those like kind of huge connection of this nucleotide and amino acid molecule. And then there's another type of hypothesis. They even like some scientists in the 1950s proposed that like in this hypothesis, there's no no oxygen, which means in the earth there's no gas. And another hypothesis they would propose like there is gas on the earth, on the earth, but that's like this atmosphere is rich in water vapor and hydrogen gas, methane, and ammonia. But this component, along with a lack of oxygen, produce a reducing atmosphere because methane and ammonia readily give up electrons to other molecules, thereby reducing them. Such oxidation reduction reaction or redox reaction are required for the formation of complex organic molecule from single inorganic molecule. So like because they don't have oxygen, they have those type of water, hydrogen and methane, ammonia, those kind of gas. Those gas can give up electrons. They are reductant, can give up electrons to inorganic molecule very easily. Then they help those kind of inorganic molecule to convert to nucleotide and amino acid, this type of organic molecule very easily. and even some like scientists, they use this kind of theory to design experimentation to test this hypothesis. What did he do? It's simulate the conditions on the Earth, right? So here's boiling water. Boiling water, they have they can generate like water vapor, water, and then this bowl have contains hydrogen gas, methane, and ammonia. And then beside this gas, the scientists will put two electrode here, because this electrode can send electrical discharge into the chamber to stimulate lightning bolts. And then there's some kind of cooling water. There's some kind of cooling water here to cool down the heat generate within this kind of chamber. And after, like, for example, he put an electrode here, because electrode is more like simulate some kind of lighting, like which happened a long time ago. And then the mixture of these four gas here, reaction can going on under those kind of electrode condition. Then after that, he took the sample from this bowl for analysis. Then he observed the formation of hydrogen cyanide and the formaldehyde. And then these two molecules are a precursor of, like, for example, amino acid and the nucleotide. These precursors combine to make a large molecule, such as amino acid glycine. So amino acid can be generated from this kind of like precursor, which generate in this kind of bowl under the electrode conditions. And then they use more experiment, and then when this like experimentation going on, after 1 week, they found out 10% of the carbon have been incorporated into organic compound. And then when they put it further, they found out the sugars, amino acid, lipid, and the bases found in this nucleic acid. So for example, they found the adenine here. So basically, from this reaction, they discovered under the lightning condition, these four gas can generate like the building blocks for nucleotide and amino acid. And let's see another one. They have another hypothesis. They even argued like sufficient organic molecule may have been present in the material from asteroid and comet that reach the surface of Earth, early Earth, in the forms of the meteorite. So which means there's some kind of organic molecule that come from another planet, and those kind of organic molecule, they reach the surface of the early Earth in the form of a meteorite. And then they found out a significant portion of a meteorite belong to a class known as the carbonaceous chondrite. Such meteorite may contain a substantial amount of organic carbon, including amino acid and nucleic acid base. So in this hypothesis, they thinking this kind of nucleotide or amino acid from another planet. Okay, they have another hypothesis called deep sea wind hypothesis. Another scientist, they proposed the key organic molecule may have originated in deep sea wind, which are cracks in the Earth's surface where superheated water rich in metal iron and hydrogen sulfide mixture, body with cold seawater. So they are thinking about maybe like in the ocean, there will be some kind of cracked in the earth crust. And then this kind of cracked in the earth crust, there will be the water here. There will be like, it's very hot here, high temperature, because this kind of high temperature here. So it's generate those kind of organic molecule. So organic molecule was generated by the deep sea wind, some kind of volcano underneath the ocean. And then this kind of generate a lot of heat, high temperature because of these extreme conditions. Organic compound can be formed. What else? Okay, let's talk about another stage. Second stage, organic polymers may have formed on the surface of the clay. There's also some hypothesis here. So three hypotheses suggest a reasonable mechanism whereby small organic molecule could have accumulated on early Earth. Scientists hypothesize that second stage in the origin of life was a period in which simple organic molecules polymerized to form more complex organic polymers such as DNA, RNA, and protein. And then a lot of hypotheses, they assume polymer with at least 30 to 60 monomers are needed to store enough information to make a viable genetic system. And then like there's some like crystallographer suggests that synthesis of a polymer took place on clay. And then he wrote a book about it. They are talking about like many clay minerals are known to bind organic molecules such as nucleotide and amino acid. Experimentally, many research groups have demonstrated the formation of a nucleic acid for a polymer and a polypeptide on the surface of a clay, given the presence of a monomer building blocks. During the prebiotic synthesis of RNA, the purine base of a nucleotide could have interact with the silicate surface of the clay. Cations such as magnesium could have bonded the nucleotide to the negative surface of the clay, thereby positioning the nucleotide in a way that promoted bond formation between the phosphate of one nucleotide and the ribose sugars of an adjacent nucleotide. So they propose those kind of things like even some kind of a magnesium could catalyze some kind of reaction, can form the RNA. And then there's another hypothesis, like they're saying like maybe polymer, RNA, DNA can also form in the aqueous solution. Okay. We'll talk about the stage 3. Cell-like structure may have originated when the polymer were enclosed by a boundary. So in this stage, the hypothesis would be formation of a boundary that separate the polymer such as RNA from the environment. So which means why we're saying this boundary is important. Like this boundary more like a lipid bilayer. So this kind of a boundary, lipid bilayer, it can separate the internal environment of the cell and the outside environment of the cell. So basically, why this boundary is important? Because it's want to maintain an internal chemical environment distinct from its outside. Like they, even some scientists, they propose some kind of characters related to this kind of boundary. So boundaries such as membrane can separate internal content of the from the outside environment. Polymer inside of the protobiome contain the information, and the polymer inside has catalytic function. These protobiomes eventually develop the capability of self replication. So basically, membrane is required to separate the internal with the outside, and then polymer, like inside of this membrane, there's some kind of polymer. They contain the genetic information, and then there's some kind of other polymer like like a protein. It can work as a catalyst, can push a reaction going on, and then the substance inside can develop the capability of the self replication. It is saying like in early stage, this kind of protobiomes cannot be precise self reproduction like a cell, but it can divide to increase the number. It can also like assume they have a basic metabolism pathways, and then the structure of organic molecule can change. And then the polymer inside can have those kind of catalytic ability to link organic building blocks to produce new polymer. And then this kind of pathway more complicated. And the ability of this type of the protobiomes to self-replicate become more refined over time. And then people even hypothesize those kind of protobiomes, which is original of cell membranes, may have existed in the lipizome. What's lipizome? Lipizome is it's vesicle surrounded by a phospholipid bilayer. So how this bilayer formed? It's like certain type of lipid. When they dropped in the water, they subcutaneous form the lipizome. So this is like a lipizome, right? It's like, for example, it's the same thing as we talked about last time. It have two layers. Outside, they are composed of lipid. Outside, they like water. So this is like yellow thing. It's they like water. They are polar phosphate functional group. Also this way, like, and then in the middle, the layers, there's like non-polar tail, right? And then how they can aggregate, become this kind of visco, because this kind of lipid, individual lipid, when they dissolve in the water, it's aggregate automatically. And then outside, and then this like outside will be flavoring water part, which is phosphate, polar part. And then inside, that's like non-polar things. They can try to get away from the water. Okay, And then they even have those kind of hypotheses claim can catalyze the formation of lipidome that grow and divide. And that's like lipidome can grow and divide. That's primitive form of self-replication. And then they found out even RNA, if it's on the surface of a clay, find a lipidome that encodes RNA are formed. So like, for example, if the RNA existing, and then this RNA can be wrapped inside of this lipidome, which is very similar to the cell structure right now, like cell structure, cell membrane. It's bilayer, right? There's a lot of like information, RNA, DNA, and the protein was wrapped inside. So, and the last stage, we are talking about cellular characters may have evolved through chemical selection, beginning with RNA fold. So they believe RNA have three key functions. RNA has the ability to store information in its nucleotide-based sequence. Due to base pairing, its nucleotide sequence have the capacity for self-replication. RNA can perform a variety of catalytic function. The result of many experiments have shown that some RNA molecule can function as a ribosome RNA molecule that catalyzes chemical reaction. And so they also found out DNA and protein are not so versatile as RNA. DNA have very limited catalytic activity. Protein are known to not going through the self-replication. RNA can perform functions that are characteristic of protein, at the same time can serve a genetic material with replication and information function. So DNA and RNA, they can replicate. They can have some kind of information, but DNA, it cannot be catalytic. Protein cannot go through self-replication. And then they have a question: how did the RNA molecule were first made prebiotically involved in a more complex molecule that developed the cell-like characteristic? Researchers proposed that a process called chemical selection was responsible. So what they mean is like when a chemical within a mixture has special property of advantage that causes it to increase in amount relative to other chemicals in the mixture, chemical reaction happens. So chemical selection results in chemical evolution. So what's that mean? So chemical evolution, which means a population of a molecule changes over time to become a new population with a different chemical composition. And the scientists even speculate initially the special property that enable certain RNA molecule to undergo chemical selection, where they are ability to self-replicate to form other catalytic function. So RNA molecule inside of these protobionts could be used as a template for prebiotic synthesis of the complementary RNA molecule. So basically, what they are saying, like, for example, they assume this kind of chemical selection happened. So for the first step, so mutation is like can provide RNA with the catalytic ability to synthesize a new RNA molecule using a pre-existing RNA molecule as template. Like, for example, this is RNA. So then a mutation will happen to one of the RNA, and then that mutated RNA can catalyze, synthesize another string, another RNA by using the old RNA as a template. then we can say this RNA have a catalytic, this is like a mutated RNA, like the old one, which didn't change, will be yellow. And then this purple one is a mutated RNA, and then it can catalyze like the yellow protein, yellow RNA to produce another RNA with the original RNA as a template. So chemical selection, what's that mean? So this is purple mutated RNA, which can work as a catalyst. It's increased because it can replicate very, very fast. Okay, and then the second step of chemical selection is like the second mutation provide RNA molecule with ability to catalyze a step in the synthesis of the ribonucleotide. So which means the purple ones will further mutate, become this green RNA. And then the second mutation is so the protobiomes have two catalytic function, self-replication and ribonucleotide synthesis. So this is a mixture of green and purple. So purple can replicate, green can synthesize ribonucleotide. So it has both of the function, self-replication and ribonucleotide synthesis, this kind of function. So they have, in this stage, they hypothesize like DNA can mutate into two types. And then those kind of mutated RNA have all kind of like a little bit like a pre-cellular function. It can catalyze, it can self-replication. It's a little bit like very similar to like pre-cells. But then that's the original hypothesis of RNA, right? But they found out that's not be true, right? Because it's different from the modern DNA, RNA, and the protein world. And so they found out like maybe the original cells, when it's developed, it's not only about like based off the function of RNA, it's maybe involved with like DNA, RNA, and the protein. They all work together, right? And then because in reality, DNA will contain genetic information. And they found out that like later on, maybe DNA was incorporated into the cell. And then RNA don't have those kind of storage function anymore. Then DNA was replacing RNA with respect to the function of the genetic information storage. And then because RNA was replaced by DNA, so RNA can have other function. For example, DNA store the information for synthesis of RNA molecule, and then such an RNA molecule could bind cofactor and have modified base or bind peptide that might enhance their catalytic function. So they found out like cells with both DNA and RNA would have a lot more advantage over just the RNA. Another advantage in DNA is stability. Compared to RNA, DNA strand are less likely to break spontaneously. A second issue is how DNA come into being. Scientists have proposed ancestral RNA molecule has the ability to make DNA using the RNA as a template. So basically, DNA is from RNA by using the RNA as a template. This is called reverse transcription, right? When it says reverse, like in the real world, DNA will be used as template for synthesis of RNA. But in this hypothesis, they are thinking like maybe DNA were made by RNA. It's the reverse. That's called reverse transcription. Okay, because this kind of reverse transcription is happening in a lot of like retrovirus. We found out that this kind of like DNA was made by RNA when we doing some kind of research by retrovirus. And then we're going to talk about like stage 4, like development of a metabolism and other cellular functions. Then from this perspective, we should think about the arranging of protein. So the protein can work as a catalyst, being a great benefit to early cells. Due to a different chemical property of 20 amino acids, proteins have vastly greater catalytic ability than do RNA molecule. So like, for example, protein, they have 20 amino acids, then which means protein have a more variety than the RNA, because RNA only have four building blocks, AUCG, right? And compared to like 20 amino acid of a protein, it's much less. So they are thinking maybe like protein can have more types of protein than RNA. So protein can have more function, catalytic function than RNA. And then protein was taking place of RNA as a catalyst, right? And the protein can also function other important tasks, like cytoskeleton protein can carry out structural roles. And the cellular member protein are responsible for the uptaking of the substance into living cells. Then people were asking how protein will become RNA. Chemical selection experiments have shown that RNA molecule can catalyze the formation of the peptide bond and even attach amino acid to primitive tRNA molecule. So basically, protein, RNA can catalyze the peptide bond formation, help to synthesize a larger protein. Like modern protein synthesis still includes a central role for RNA in the synthesis of the polypeptide, right? When like, for example, nowadays, if you want to work in the lab, how to like, for example, synthesize the protein outside of a cell, people always involve the RNA as a catalyst to synthesize the polypeptide. And then how did it do? First, mRNA will provide information for the polypeptide sequence. Like for example, RNA can have the information for the protein, right? The sequence of a nucleotide decide the amino acid sequence. And then tRNA, transfer RNA, can work as an adapter for the formation of the polypeptide. What that mean? So different type of tRNA will grab different amino acid in the cells. And then by grabbing the right type of amino acid, it can attach to the old polypeptide chain to form the large proteins. mRNA and tRNA, it's when we talk about the protein synthesis, tRNA and mRNA, both of them will be involved in the synthesis of the protein. And then ribosome. Ribosome is a big, it's a tRNA, it's RNA and the protein complex. It's like an organelle. It's provide the site for protein synthesis. When we're talking about the protein synthesis, we know in a cell, protein synthesis happened in the organelle called the ribosome. Ribosome is a big, complicated organelle. It's a combination of RNA and the proteins. And rRNA molecule in a peptide transferase complex within the ribosome. So basically, this is ribosome. Ribosome have RNA, and this is peptide transferase. It's a protein. So ribosome is composed two part, rRNA and the peptide transferase protein. And this ribosome can act as a ribosome to catalyze the peptide bond formation. Okay, and then the analysis of translation modern cells is consistent with our evolution history, in which RNA molecule was instrumental in the emergence of formation of the proteins. So this fourth stage describes how the cell was developed, like at the very beginning. There's a lot of hypotheses, like for example, how this small molecule inside of a cell, how it's formed, because long time ago, there's like extreme weather condition. This kind of extreme condition can convert very simple gas into the building blocks of the protein RNA. And then after those kind of building blocks was formed, and then the RNA was formed. Okay, and the RNA is a big molecule. It's keep changing. It's keeping mutating. Can work as a catalyst. Can work with like can self-replicate, right? And then with the time, DNA and the MR and the protein be generated with the help of the RNA, and then it enriched the content within the cell. So now cell have DNA, RNA, and the protein, which more represented, which is more like similar to the nowadays real cell. And then this RNA, DNA, protein, and the small molecule all can form all kind of organelle in the cells, within the cell. And with time and development, and then the real cell happens, the life happens. That's how it's talked about in this kind of structure, in this chapter.